Beep-a-doo, beep-a-doo. Good morning, everybody. Blimey, 9:30 already. But thankfully, this time I have a cup of coffee. Okay. All right. We've got all of 4 students here today, which doesn't bode well. Okay, so dum-de-dum-de-dum. We might as well get started. So what I think would be a good idea first up is if anyone has any questions about the general mechanism of DNA replication, right? The stuff that I covered in that short extra video on Thursday, kind of after I had to leave to take my wife to an appointment. Anybody have questions on that? Anybody? Otherwise, I'll just assume that you understand it all. All right. Well, okay. So, oh, hey, look at that. I was doing some research and studying of my own to figure out how DNA polymerase alpha works, which is something that we'll be covering today. Okay, so essentially what I did in that little extra supplemental bit of the lecture, which was posted to YouTube last week. Sure, what was the day today? Tuesday today, yeah, that's right. Would be last week. Was to essentially go over the basics of DNA replication in prokaryotes, which are in some ways a little simpler than eukaryotes, right? A little bit more straightforward to understand, right? And so we're really talking about initiation, unwinding, right? And the roles of topoisomerases in that process, which are really, really important. Again, DNA gyrase is the prokaryotic name for topoisomerase. If you look at stuff of eukaryotes, it's always called topoisomerase. They are functionally the same thing, right? DNA gyrase is just one of many of a class of proteins called topoisomerases. Next thing, and this is going to be important because eukaryotes do this differently, right, is the formation of a RNA primer, right, using an enzyme called primase, right? So these are all kind of steps and proteins that you're going to need to know, right? Because I will be testing you on them, right? Because if you don't know them, then it's kind of hard to explain how the whole thing works, right? And its role is to create a short RNA primer, most 20 nucleotides long, to serve as a starting point for DNA synthesis with DNA polymerase. Quick refresh on Okazaki fragments. Then a little bit of an overview of the different types of DNA polymerase in bacteria, right? They are different in eukaryotes. And then essentially the process of removing those primers and resealing the backbone, the sugar phosphate backbone, right? So that the new daughter strand is continuous. And this is also something that differs in eukaryotes, right? So it's worth understanding this so you don't get confused between how prokaryotes and how eukaryotes do it. Let me just, oh shoot, let me just see. I should have done this before. Hang on, get off of it. Just need to double check where I ended up on, when was this? This was last week. Sorry, I should have done this before starting class. Dum dum dum. It's always weird watching myself do stuff. All right, so essentially, there we go. Now I know where I'm at. Oh, isn't that weird? It's like looking at myself, looking at myself. Okay, so we ended up pretty much around here. So what we have left is proofreading, right? How to ensure the accuracy of DNA replication, which is crucial because of the vast number of times your cells will divide. This not only applies to eukaryotes, but also prokaryotes, because if you essentially every bacterium, what's that dog doing? Will be the clonal offspring of its, or clone of its parent, right? And bacteria can double very, very fast in number. Some E. coli can do it as quick as once every 20 minutes, right? So over a period of a few hours, you can go through many rounds of cell division, right? And so you want to make sure that you don't accumulate hordes of mutations along the way. And that's even more critical for a multicellular organism like eukaryotes, where you're the sum of essentially thousands of rounds of, or thousands of cell division events, right, that generate, what you are as an organism. And so you can't have some of the cells in a particular tissue having one set of instructions and another set having a different set due to mutation. So there's essentially a series of kind of different steps. Hang on a sec. Amor, I think one of the puppies, Amor, hey love, I think one of the puppies is digging into something. Thanks. One of our foster pups has a digging fetish, and so we have to kind of make sure he isn't digging her way through the bottom of the couch or something. So essentially, there's three different kind of ways in which accuracy is ensured, and you get to essentially in 1 in a billion to 1 in 10 billion accuracy rate, right? I wonder what's a billion. Yeah, billions is 9. Yeah, so it's between 1 in a billion and 1 in one error, sorry, in a billion and one in 10 billion, somewhere between those two. Right, so if you consider bacterium, which has, kind of what about less than a million base pair genome, you're pretty much sure that you're not going to be creating mutations every time you divide. Anyway, so there's 3 different. Yeah, go on, give me a second. I've just got to go and sort this out. Sorry about that. They were chewing up a wicker basket underneath one of the tables. All right, which is now lots of bits of wicker basket. Okay, so essentially there's 3 steps. One is the inherent accuracy of DNA replication, which is based on those hydrogen bonds and the shape, right? So you have an inherent inclination towards accuracy, right? Based simply on which nucleotides will base pair with which nucleotides, right? So that's essentially an inherent property of DNA, right? Which makes its replication fairly accurate. That's kind of about 1 in, oh shit, that didn't make things any better. About 1 in 10,000 bases copied will be wrong simply by that mechanism alone. That's a little bit better. The thing is, I can't see my screen. My eyesight is so bad. There we go, more or less. See our one eye at least. The next step, right, is essentially a function of the polymerase, the DNA polymerase, which is able to correct its own mistakes as it goes through the process of adding nucleotides to the new daughter strand, right? This is called proofreading activity, right? Which, hang on. Essentially involves it stopping once it realizes it's made a mistake, going back and cutting that erroneous base off and then replacing it with the correct one, right? So this is very much analogous to you as you're, typing along, realizing you spelled something wrong, going backspace, 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 put the right letter in, and then you can continue writing your essay or whatever. That's essentially what DNA polymerase does as it synthesizes the new strand. And that will get you probably about another 1 in 100 improvement, right? So that gets you up to about 1 in 10 million, give or take, errors. And then the final bit is essentially what happens after DNA replication has occurred, but, and this is a very important but, before the new DNA strand has been methylated, right? Methylated, essentially it's a bunch of small tags which are added to DNA, methyl groups. And essentially the daughter strand does not have those for a short period after the end of DNA replication. And so because it doesn't have those marks, post-replication mismatch repair can recognize which is the daughter strand, i.e. the unmarked one, and which is the template strand, the marked one, which is presumably correct, right? And so it can then go back and fix that error before those differences between the template and the daughter strand are erased, right? After that point, you're kind of screwed because you can't figure out which was the template strand and which was the daughter. So, which one has the wrong base in it, right? There's no way of telling. Okay, so this proofreading is found on DNA polymerase 3, right? So this is the main synthesizing enzyme, DNA synthesizing enzyme in bacteria. In eukaryotes, there's 2 which do the same job, called delta and epsilon, right? One for the leading and one for the lagging strand. In bacteria, we just have the one which does both leading and lagging strands, right? And so essentially its job is when it realizes it's stopped, well, once it realizes it's incorporated the wrong nucleotide. And that's, I don't actually even know why that happens, just random chance, I guess, right? And it's already made that phosphodiester bond, it stops, essentially backpedals a little bit, right, 1 base, and then it cuts that phosphodiester bond, it hydrolyzes that bond itself. This is called exonuclease activity, and it can only do it from the 3' to 5' direction. Pardon me. So essentially it's able to chew off a little bit of the end that it's just made, but it can't digest DNA anywhere else, right? So it's very, very specific to this situation. And so as long as it catches it in time, it can walk back, cut that bond. That nucleotide is now ejected, right? Poof. And the correct one is allowed to come in, which in this case would be a thiamine. The bond is recreated, and then DNA polymerase continues off on its way, right? And so this reduces the errors quite a fair bit, essentially another 100-fold. But it can only happen if the DNA polymerase detects that it's made a mistake at the very end, like the very last one that is added. If it detects it's made that mistake, it can fix it. If it adds another base after that, say a guanine, right, to match up with this cytosine, and makes that phosphodiester bond, it's done, right? That error will be incorporated, right? Because it can only cut off one base. It can't cut off two, right? So it has to catch that error immediately to be able to fix it. If not, that's going to have to be caught by a process called mismatch repair. Right, so this is in eukaryotes, but it's a pretty similar process in, sorry, this is in prokaryotes. It's a very similar process in eukaryotes. And essentially what happens here is that a series of proteins recognize this mismatch, right? And so that mismatch typically creates a change in shape of the DNA strand, which is actually shown here as being a little bubble, basically, right? Because these two bases don't bond properly. They don't actually fit together very well, right? And so they create a change in the shape of the DNA, which is recognized by other proteins, right? And so it's a process again, right? So that mismatch is recognized, right? And essentially that protein that recognizes it recruits a bunch of other proteins. And what they do is they find, in this case, the next A, right? And they will cut. So you can see there's a methylated A here. It'll go for the next one, find that it isn't methylated, and then realize that, hey, this is the daughter strand. And then it'll cut the DNA. It'll cut a single strand, create a nick, right?
Dr. Crick, I'm sorry.
Hello.
Oh, so what's again meant by methylated? What does that mean?
Okay, so essentially, here I'll do it on the board a little bit easier. So, oh man, where my pen's gone? Typically, it's on C's, but can also be on A's. Let's say you have this will be G. Actually, let's do this in blue because this will be the daughter strand. G, T. So if this is the template, and this is a daughter, all DNA ends up with essentially what are called epigenetic tags, right? So these are, kind of think of them like skin tags in a sense, right? And these are, the fancy word is methylation marks.
So they all have like a methyl group? They're all supposed to have a methyl?
Typically, no, not all of them. In fact, actually, it's typically what are called CPGs, which are C's. Actually, let's do it a different way. This is going to be better. Got to do things accurately, unfortunately. There you go. May seem like pedantry, but I don't want to teach you the wrong stuff. Typically, you have them where you have C's and G's together, right? And the C's will be methylated. And essentially, those are, I mean, most of them will be, not always. It's not a kind of a given fact that you'll always have all of the C's and the A's methylated. It varies depending on activities of other enzymes. Actually, in many ways, it's a unique imprint on you, which reflects the experiences you've had as an organism since birth, right? And these marks, these epigenetic marks, are what vary between identical twins. And so they're influenced by environmental stuff. So if one twin smokes and the other one doesn't, for example, you'll get different marks in one twin versus the other. Obviously, smoking's not the only thing that does it, but there's an awful lot of other stuff as well, right? And that's basically why twins start looking identical twins start looking less alike as they grow older. So if you look at young children that are twins and they're like, you can't tell them apart. If you look at 40, 50-year-old twins, they'll look very, very, very similar, but you will be able to distinguish between them, right? And that's in part because of the marks that they've accumulated. However, there is a baseline degree of methylation, right? These marks. So you don't change the actual base, right? The information is the same. The nitrogenous base stays the same, but the little tags that you have on the outside can change, right? However, these marks only exist on essentially old DNA. So if you've just copied this template strand, you will have copied the information, right? So now you have your daughter strand, but it takes a while for the methylation enzymes to come along and add the marks. Like, for example, you would have one here at least. But because it takes time for that to happen, there's a small window after finishing DNA replication, but before those methylases come along, in which you can tell the template from the daughter. After that point, when this gets methylated, like so, how can you tell which strand is which?
So the old strand's gonna have more.
For a short period of time, yes. And that's not because the old strand is somehow different from the new strand. It's simply because, over here, you'll have your, I think it's called DAM methylase, right? And it's going to be kind of trundling along, adding methyl groups everywhere that there should be one. That's why if this mismatch repair does not occur quickly, right after the end of DNA replication, it won't occur at all. Because it relies on the lack of methyl groups on the daughter strand. Does that make sense?
Yes, thank you.
Yeah, good. Thanks for the question. And so that's in a sense why one of the reasons why DNA replication is very, very accurate, but it's not 100% accurate, right? Because some of these errors will slip past the DNA polymerase, right? Its exonuclease activity is going to catch most of them, but not all of them. And then B, most of the rest of the errors that are created, that are missed by the DNA polymerase, are caught by the mismatch repair mechanism. But if those don't get fixed quickly enough, methylation occurs, and that mutation, that change, is now incorporated into the genome of that organism. So the next time that cell replicates or divides, you're essentially going to have what was the template in the daughter, right? That now becomes its own chromosome because it'll get copied. This one becomes its own chromosome because it gets copied, right? And so that error, if you have an error in, let's say, around one of my knuckles, right, that's incorporated into that daughter strand and it isn't caught, then every cell that descends from that lineage on that side will have that mutation in its genome. This one will be fine. Right, so this has to occur real quick because otherwise it doesn't work. And if it doesn't work, there's nothing else. No, nothing else that will actually fix that mistake because there's no way of knowing which is the mistake and which is the correct base. You can't tell. And that's all down to that methylation. Once that methylation is complete for the daughter strand, there's no way of telling her which one was the daughter strand and which one was a template strand. You can't. They essentially are the same. Okay, so once that error has been detected and these proteins recruited, right? Basically, the ones which have the endonuclease activity, right? They can cut in the middle of a strand, find the next GATC. This is just like a motif, essentially, and they will cut the backbone just to the right of that GATC, right? And you're going to get these combination of bases just randomly throughout the genome, right? Doesn't mean they do anything. They're just it's just a random collection of letters, right? Which you'll get just by chance. However, what then happens is that they will use another protein will use that nick, right? So now we have like a free end of DNA, right? If you remember the whole exonuclease activity of DNA polymerase only works at the end of a piece of DNA. We have the same here. So we now have a free end, and now an exonuclease chews up that daughter strand, right? Basically kind of gets rid of all of those bases that are part of that daughter strand until it goes past that error. And so what we're doing, we're not actually just taking out the error, right? We're taking out a whole bunch of DNA, including the error, right? And this is actually a common feature of DNA repair. You see it with fixing thymidine dimers, for example, which you get from UV exposure. And then as soon as that bit of single-stranded DNA kind of appears, now you have another DNA polymerase comes in and it will fill in that gap, one hopes, with the correct bases. And as soon as that occurs, your DAM methylase, the enzyme which adds the methyl groups, will come along and methylate the daughter strand. And then it's fixed. Okay, so I'm going to moving somewhat quickly on. Actually, before I go on, anybody got any questions? Just make sure I catch anything before moving on. Okay, so in eukaryotes, let me get up the chat. Where are we at? Euks have several key differences in DNA replication. Some of these are differences in, what does what, essentially, right? Small, kind of subtle differences. Some of them are pretty big. So one of the first ones and biggest ones is, pardon me, eukaryote DNA essentially is chromatin, right? Which is a mix of DNA and protein. And those proteins are called histones, right? So that's something that you got to kick off, right? And also make more of those proteins, because now you're going to have twice the amount of DNA, so you have to put them back. And just, Tanya, just to add an extra level of complexity, those histones are also marked with different tags on them. And so basically this shit gets complicated pretty fast, right? And so obviously you're going to have half of those histones are going to be marked because they're old ones, and then half of them are going to be new because they've been freshly made and aren't marked. So it just gets all kinds of complicated. We'll get into that maybe later when we talk about epigenetics. What else? Two, euks have linear chroms. So that basically equals the end replication problem. Right, which is something that bacteria do not suffer from because they have circular chromosomes. Right, and we'll talk about what that means and the consequences of that in a wee bit. It's actually really, really interesting. I think so, at least. What else? Different enzymes. If I can spell enzyme. Hey, I'm no chemist, right? Do different things. So no DNA primase. That's prokaryotes. Instead, we have DNA pol alpha, which is pretty funky. Also have a DNA pol for leading, which is, oh shit bits, which one is that? I think that's delta. Where?
I was gonna ask, is this where it makes like a compound with all like the alpha and the beta and something like that? That they make like this bob?
Well, it definitely is going to be a complex. Let me just finish what I was writing before I forget. Oh yeah, epsilon is the leading. And finish off. Primer removal is done differently, which we'll get to as well. So it's complex. Tanya, it's the easy answer with eukaryotes. Everything's bloody complex. It's always complex, right? So DNA polymerase alpha, this is what I was reading up this morning because I had interesting thought from class yesterday. So DNA polymerase alpha is not a holoenzyme complex. It is a multi-subunit protein, right? And that seems like a bit of a semantic difference, and in some ways it is. Holoenzyme complexes typically are aggregates of proteins which come together to do a particular job. So RNA polymerase and transcription is a classic example of that. And actually the whole kind of helicase, DNA polymerase, DNA primase deal in bacteria is another holoenzyme. This, however, is one single protein. It just consists of separate proteins that come together and assemble. Right, so this doesn't function, none of these parts can function on their own. You have to have the four together. And in DNA polymerase alpha, 3 of those essentially do the same job as primase does in bacteria, right, which is RNA polymerase. And those 3 are kind of grouped together with another subunit which does a short bit of DNA synthesis. Right, so essentially, kind of like an easy way of putting it is eukaryotes are always more complex. They just are. Which also makes them really fun to study as well, in my opinion at least. Good question, Ali. I'll get to that. You'll see what a doozy it is in a little bit. Okay, so just make sure I've got time for that. I'm going to kind of rush on. This isn't something you need to worry too much about, only that there are 8 histones in a complex, in a nucleosome, and there's another one called histone H1, which kind of glues things together and creates a spacing between them, right? And so eukaryotic DNA is always complexed with protein. We also have, this is essentially a summary of differences, right? And one of the weird things about DNA replication in eukaryotes is we have one DNA polymerase that starts the job, right? This is DNA polymerase alpha. It synthesizes the RNA primer, which is weird, right? And then 2, it does, I don't know, I think it's like between 30 and 100 bases of DNA nucleotides. Then it falls off. It's like, oh, I'm all out of juice. Sorry, chaps, can't do any more. Right? It is not a long distance runner, right? It's like maybe 100-meter sprinter at most, right? And when it falls off, depending on which strand it falls off of, another DNA polymerase comes in and finishes the job, basically does all of the rest, right? And so that's either epsilon for the lagging strand, in which case it will do like plink, fall off because it's an Okazaki fragment. Or if it's epsilon, it will just keep going for miles of DNA, right? And these 2 also have that exonuclease activity, right? So these epsilon and delta can do the whole proofreading thing. So really, you don't want alpha hanging around for very long because it's not going to fix its own mistakes. Its job is really to get the party rolling, and then another one comes in and finishes and just goes the distance for the rest of it, right? Also, getting rid of primers is a different deal, right? So in a way, this is kind of more analogous to mismatch repair. that we talked about earlier. So if you remember in prokaryotes, right, we have one DNA polymerase 1, handily enough. And its job is to both chew up the RNA primer and synthesize the DNA in the gap, right, to fill in that gap that the removal of the primer created. So it has kind of a dual function, essentially. In eukaryotes, it's done quite differently. So here, essentially, the DNA polymerase, this is delta, because we're talking about the lagging strand. This also happens on the leading strand too. It just doesn't happen very often, right? Because in eukaryotes, you can have really, really long leading strands, but you have very, very short Okazaki fragments. So that's why we're using DNA polymerase delta here. It's not that it only occurs with the lagging strand. It just happens a hell of a lot more often, right? That's why we use it. Okay, so essentially what happens is this DNA polymerase delta, it will kind of chug along until it hits essentially the beginning of the fragment that was synthesized previously, right? Remember, the lagging strand is kind of synthesized backwards, walking forwards, right? And so essentially this is the new Okazaki fragment. This is the one that was done before that, and it will kind of go backwards until it hits what essentially has already been made, right? And when it does that, it pauses. It doesn't fall off. It just kind of like sits there, scratching its head. What's going on here? And it waits for that RNA primer to be removed. And so that's carried out by what are called single-stranded replication protein A, right? Single-stranded DNA binding protein replication protein A, there, or RPA for short. Its job is essentially to lever off this RNA primer, right? So it kind of goes along and just like kind of levers that RNA primer off and a bunch of DNA. Now you might think, well, why would you want to kind of undo what you've already done? Well, it doesn't really matter because the DNA polymerase is there ready and waiting to fill in the gaps, right? So it doesn't actually matter. What really matters is you get all of the RNA primer, right? So you can be a little bit sloppy with shooting past the RNA primer as long as you make sure you've got all of the RNA primer, right? There's not going to be any RNA remaining. And so once RPA has kind of kicked it off, then we have another enzyme come in and we just snip it, right? I don't know what the analogy would be for that, but that whole chunk of RNA and DNA is removed. And now obviously DNA polymerase delta is like, oh, sweet, kind of free road ahead. And it will just start up again and fill in the gap, right? So this is very different from DNA polymerase 1 in prokaryotes. Right, just let you kind of catch up and then we'll get on to the end replication problem. I'm just going to take a picture of this, just in case I need the board. Ah, my wife's been tidying up. Here we go. So I now have a more professional looking office. I just can't find anything. Okay. Okay, so the end replication problem is a doozy. This is an American word that I picked up since I came to the US. Didn't know what it meant. I thought it might be like a floozy, but obviously it isn't. All right, we don't want to talk about floozies. Yeah, do you know what floozy means? No? Oh, well, probably
I
better
don't
not
know
to.
what floozy or doozy. I'm just like, okay. It means like weird, I guess.
Doozy is
My,
like a big one.
I
Oh,
don't
that's
know
a
what it is.
doozy of a problem. That's like a real big issue. I think, at least. Hopefully an American will correct me if I'm wrong. Anyway, floozy, we'll just not worry about that. Anyway, moving swiftly on. So the issue is linear chromosomes. So think about this. We have RNA primers used to get DNA replication going, right? We have to get rid of those RNA primers once DNA replication is, nearly complete, at least the copying of the DNA. To get rid of those RNA primers, we need to have somewhere a DNA polymerase can continue replication of DNA, right? That can only occur when DNA polymerase has a 3' OH to add a new nucleotide to. That's the whole point of primers, to get around that issue. So what happens when this primer is right at the very end of the chromosome? We can remove it because we don't want RNA in our genome, right? For shizzles, basically, right? But there's nowhere for DNA. So essentially, if you kind of put your hand up, right, to the screen and cover up like the left-hand side of that graphic. Come on, Tanya, let's see. Come on, put your hand up. Come on. There you go, right? That's what it's like at the end of each end of the chromosome that's being copied. So there's no 3' OH. There's nowhere for DNA polymerase to fill in that gap. So that means that every time you copy that chromosome, you will have a 15 to maybe 20 base pair, it kind of depends. A short stretch, which is single-stranded, right? Essentially looks like so. You might think, yeah, well, so what, right? It's only 15 bases, it's not very long. Next time this cell divides, this red strand will be the template for one of the new chromosomes. The gray strand also, right? Because it's semi-conservative DNA replication. However, this red strand will be a little bit shorter. Same problem occurs again, right? This red strand now, one of the copies of that will have a gap left at the end, and so on and so forth. So every time your chromosomes are copied, they get shorter because there's no way to fill in that gap left by the removal of the primer. Okay, so that's the end replication problem. And this is an interesting problem because you're probably thinking, well, what are the consequences of that, right? So if we had, let's say, if we had a gene right at the very end of that chromosome, over time, it will get eroded, much like the cliffs on a, a stormy, not Gulf beach, like say the Atlantic or the Pacific. You always hear about the Pacific Coast Highway in California, like falling down and stuff, right? So essentially that gene will slowly get eroded, right? And at some point it will lose function, right? So whatever that is, it will stop working over time. And that would be pretty terrible, right? You would essentially lose information and the ability to produce certain proteins as you age, right? Which would not be good. And some types of cells are more prone to this than others. Like some types of cells replicate constantly, right? Some don't. Neurons, for example, they're never going to replicate again once they become neurons. Intestine, like all the time. It's like this constant conveyor belt of cells going from the bottom of your crypts, kind of up to the top, and then they get like chewed up or blown away or whatever, and then they get replaced, right? So depending on which cells, it's actually crazy. It's really, really interesting. It's actually crazy. Some of the cells, I mean, skin obviously is a classic example because you can see it, are continuously getting replaced. And so we get around this couple of different ways, right? One, yeah, totally. That's exactly what this is all about. So to deal with this problem, there's like a sticking plaster solution, right? And that is we have long stretches. Actually, I'd have to look into how long. Maybe a few thousand base pairs, right? Of DNA at the ends of each chromosome called telomeres, right? Exactly as what Tanya said. So telomeres are long stretches of god damn it. Oops, sorry, I shouldn't swear. Repetitive DNA at the ends, at each end. Even then I couldn't spell it right. My typing sucks this morning. Right, and their job is essentially to act as buffers, right? So they can be eroded without affecting function of any genes on that chromosome. Right, so they don't encode any information, right? They are purely non-coding. So they act as buffers to kind of, I don't know. Yeah, they just act as buffers. So that end replication problem still occurs, but it doesn't as yet make a difference. And so Tanya, you're right. Essentially, my chromosomes will be shorter than yours, and yours will be shorter than those of your children. And your eldest children will be shorter than those of your youngest children, right? Your chromosomes shorten with age. Ooh, yeah, stress is a terrible thing. Oh, there you go. I mean, there's all kinds of funky stuff with stress, like hormonal as well. Cortisone? Yeah. Cortisone is a classic stress hormone that affects all kinds of different things. Yeah, humans are complex organisms without a doubt. It's really actually hard to figure out what causes what and how. But yeah, essentially, and so this cannot go on forever, right? Because at some point you're going to run out of buffer. And so at some point, replication has to stop. And this is called the Hayflick limit. And that basically says a cell can only divide around 60 times. Once it has hit that limit, it is essentially shut down. It cannot replicate any further. And in many ways, that actually leads to something called senescence. And senescence is essentially cellular aging, right? The cells don't die, they just become less functional. And that also increases with age and presumably stress and other things which accelerate the process of aging. So here's another question to make you think. What cells never age? What cells are able to replicate indefinitely? There are two types. Well, yeah, two types. Immortal cells, in a sense. Actually, this caused a breakthrough in biomedical science and a really major ethical issue with informed consent. Just in case any of you have heard of a woman called Henrietta Lacks, well, you should do. Almost all of the biomedical discoveries in the last 50 years were made using derivatives of her cells, which she didn't consent to them being used. So Henrietta Lacks died of cancer. So yeah, so there's actually really good. Tanya and Gabriel, you're correct, just in slightly different ways. So stem cells, right? And really the cells which produce gametes are a type of stem cell, particularly in males, right? Because in females, all of your gametes are made before you're even born. In males, you continue making them throughout your whole life. Yeah, Kim, HeLa cells. That's exactly it. That's why they're called HeLa cells. It's named after the person, Henrietta Lacks. I can't remember. I think it was ovarian cancer that she died from. Really young, which was pretty tragic, like 45 or 50. Anyway, so stem cells can divide indefinitely, right? That's how you're able to keep producing hair, right? Throughout your whole life, and skin and gut cells and various other epithelial cells, right? Um, and cancer cells, pardon me. Because if you imagine, right, cancer is the uncontrolled growth and out of context restarting of development, right? So those cells divide over and over and over and over and over again until you can end up with a equal body mass of cancerous cells, right? The only way they can do that is if they can escape the Hayflick limit, right? And the way that we do that, and in stem cells, is through the activation of an enzyme called telomerase. And so essentially what telomerase does is oh, come on, get out of the way. It rebuilds, sorry, I'm trying to move my chat box. It rebuilds those shortened telomeres. So that's why stem cells never hit that limit, because they don't erode their telomeres. They're constantly being rebuilt. And that's also how cancer cells evade it, because they reactivate that telomerase, even though they shouldn't, right? Because it should be like totally shut down, right? This is one of the mechanisms, one of many, against overproliferation of cells, right? Is you shut down the telomerase, and so any cells that do proliferate when they shouldn't, because there's a whole bunch of other checks and processes as well. But even if they do, they should hit that Hayflick limit and stop, right? Somehow in tumor cells, they get around that. And so the big breakthrough for HeLa cells was they were the first immortalized cell line. Before that point, you could isolate cells from humans to study, but they would only divide for a certain number of times before they would stop. They would senesce because of that Hayflick limit. HeLa cells did not. And so those have basically, I mean, thousands of labs around the world are using descendants of the cells that were originally taken from that woman in the, shoot, I don't know, mid 60s, 70s, something like that. Which is kind of crazy. Bit of a major issue because her family have never received any kind of compensation for that. She never even consented to it, right? She didn't know what was going to happen. So that's kind of like one of the like real chunky issues of biomedical science. Anyway, we all benefit from it, but which is good, but she didn't actually consent to that happening, which is bad. So it's kind of a real tricky kind of ethical issue. So anyway, that's kind of like a fun thing to talk about, particularly like how cells escape that limit, right? Because it's just neat. Anyway, so what I'm going to do for the last actually, anybody got any questions before I move on? Because we only have one thing left to do today in our whirlwind lecture. Nope. Cool. All right, so the very last thing to do is talk about homologous recombination, right? And so this is one, it's, I always found, I don't know why I keep doing it. I've got this habit of like online teaching of like counting things off with my fingers. I don't know why. Anyway, one thing is that this is critical in prophase 1 of meiosis, right? This is what allows that kind of reshuffling of alleles in daughter cells, right? And actually, the second thing this is really important for is it allows fixing double-stranded breaks, right? And double-stranded breaks, or DSBs in the, in the lingo, are catastrophic. So if you, and they're typically what you get from ionizing radiation, like gamma radiation. So high energy radiation, gamma, X-ray, maybe some of the other radioactive decay types, maybe beta particles. Basically, if they hit your DNA, they will break it and they will snap it in two, right? So you have two ends kind of flapping around. And that is absolutely terrible, right? Particularly if you have more than two breaks, because how do you know which end goes where, right? There's nothing on the end of a bit of DNA which says, I go with this bit, right? And so you can actually have like huge whole scale chromosome rearrangements if you are exposed to, say, gamma radiation, right? So actually one of the main causes of death and the symptoms of radiation poisoning are typically seen in cells which replicate a lot. Gut cells, skin cells, hair cells, right? Because those accumulate large numbers of double-stranded breaks and those cells just die, right? They cannot divide anymore because they just have too much damage, right? And so homologous recombination, if you're lucky enough to be in the right stage of cell division, you can use this to fix those double-stranded breaks really neatly. And actually, there's a bacterium which is called, jokingly, Conan the Bacterium, after Conan the Barbarian. Probably none of you have ever seen that film. It's kind of sad. Yeah, well, you should. It's really stupid, with Arnold Schwarzenegger in it. He's like a big guy with a sword. They're able to tolerate enormous levels of radiation, about 800 times what would kill us in about an hour. And they can grow under that because they're able to do a lot of this. Something radiodurans is its name. Anyway, I'm getting on a sidetrack and we don't have time for that. So essentially what happens in homologous recombination is, one, these two chromosomes are lined up perfectly, base by base, because you do not want to have one end up longer and the other end up shorter, right? That would also not be good. Right, so this has to be perfect, like perfectly matched. So you don't gain, pardon me, or lose any DNA during this process, right? So the first step is that you break either intentionally in meiosis or accidentally because you got hit by, gamma radiation, a double-stranded break in one of those strands, right? And it's double-stranded because it is both backbones are broken in the same place. Right? It's not one of them. It's not a nick, like, with mismatch repair, for example. It's a double-stranded break. Then the first step is to chew back some of this, right? Much the same as in mismatch repair, right? So some of those nucleotides are removed from each strand. So essentially you end up with 2 bits of single-stranded DNA. This is really important because these bits are going to basically match with the other intact strands. Right? And this is actually called strand invasion, which is kind of like a weird kind of term to think about. But this free strand here will invade its matching double-stranded bit of DNA on the other side, right? So these are kind of lined up. And so basically it kind of goes, yeah, and forces its way in, right? Displacing one of those strands. And it can do that because it can base pair. Remember, these are perfectly lined up. They're not like that or like that. They are, yeah, that's perfect. Exactly lined up. And so this broken strand, right, it invades and matches. And essentially what will happen is that we will start DNA replication again. Right, and so this free 3' end will act as the start of DNA replication. So DNA polymerase will come in and continue the synthesis of that 3' end, that blue strand. Right? Does that make sense? So essentially now you're going to have a complex of like 3 strands of DNA, which is super weird, right? You're going to have 2 paired together, which is going to be one original red strand and one broken blue strand. And you're going to have a third strand, which is going to be this displaced original red strand down here. So this is going to continue on for a little bit. And at the same time, now we're going to use this displaced strand. It's going to pair up with this bit down here. Right, so now you should be able to get base pairing between this displaced red strand, right, and it now overlaps the area of single-stranded DNA for this broken strand down here. Does that make sense? No? Does this make sense? Right, so now you can, right, so we've got our invasive strand, right? It's kind of wormed its way in here, right? So this is now bit of double-stranded DNA, right? These match up perfectly, right? Because they are essentially the same, DNA. But these 2 strands match perfectly and are being replicated this way. These 2 strands will also match perfectly, right? Again, because they're exactly complementary to each other. That's how this works, right? It's how it can work. And as soon as we now have a bit of double-stranded DNA there, right, with that displaced red strand and with that broken end of the blue strand, we can now get the same deal. We can now get DNA polymerase binding and will start extending that broken blue strand. Right, so basically the steps are break it, invade it, and extend it. Right. So we've broken it up here. Yeah, that's right. Then you invade it here, right, to create that double-stranded DNA between the unbroken and the broken strand. That forces this other strand out to the extent that it will then be able to pair with the other broken strand. And then both of those ends, right, we've got a 3' end here, a 3' end here, will be extended until they can be sealed up with the 5' end of the broken strand. Right, this is going to continue all along the bottom here until it gets to the 5' end. This end is going to continue all the way along up here until it gets to this 5' end. And as soon as they are done, they will be sealed or ligated. And you'll now have this weird mix of double-stranded DNA, which is attached to other bits of double-stranded DNA, right? So we now have kind of intact original on both sides, 2 red and 2 blue, 2 red and 2 blue. And then we'll have a region where we have a mix of red and blue and red and blue. Right? This creates what are called holiday junctions, right? Essentially where these strands cross over from the original intact strand to the opposite chromosome on the other strand. Now, obviously, this cannot continue, right? Because if that were the case, you'd never be able to pull these apart during metaphase 1, right? They'd be like constantly stuck together, right? And so these holiday junctions have to be resolved, right? They have to be cut. And so this is actually what they look like in, and this is what scanning electron micrograph, maybe, might be transmission, probably transmission. I'm not very good at electron microscopy, right? So this is a high resolution image of actual DNA that is in the process of going through homologous recombination. You get a classic kind of X-shaped, right, structure with a bubble in the middle. And that bubble consists of these two kind of hybrid strands that are a result of this replication here and this replication here, right? And these junctions have to be broken. This isn't like super well understood, I have to say, but depending on which way they're broken, you can get kind of different combinations of DNA depending on where the actual site of crossover is. It doesn't make any difference if for what allele combinations you'll get on the outside, right? Because in the end, you're going to swap alleles, but it does make a difference as to what the DNA looks like and where that break is. Don't get too worried about the difference between these two. It's not super relevant to your understanding of the process. But just realize that before metaphase 1 can occur, you have to break these junctions, right? You have to cut the DNA and then stick it back together again. So you have one intact chromosome on one side and another intact chromosome on the other side, which is still going to be held together at their ends by cohesin, right? That's what will break down in metaphase 1, but not at the centromeres. That happens in metaphase 2. But as you can see as well, right, this is the kind of context that we're talking about. This is more in terms of meiosis, prophase 1. But if you have a homologous chromosome kind of present that you can use as a template, if one of them does get broken, right? So typically this only really works during a specific part of cell division, right? When you have sister chromatids that are identical, you can do this if you get these double-stranded breaks, right? So you can fix that if you get accidental double-stranded break. If you have an exact template hanging around that you can use to fix that, you can fix it perfectly. If you do not, the only way to fix that double-stranded break is through a process called non-homologous end joining. And really the kind of informal way of describing that is a Hail Mary, shit, let's stick this back together and hope we got the right end, right? And that's basically taking any free end of DNA and just sticking it back onto another free end of DNA. And so that's actually how we have gotten, in many ways, the chromosomes that we have. We had accidental events in the past sticking this bit of chromosome onto this bit of chromosome, and now you get a bigger, newer chromosome. It's not what you really want, because typically when you do that, you don't do it right. And you'll get all kinds of funkiness. You'll get different genes fused together. You'll get genes that are split apart. You'll get like crazy, crazy, crazy stuff. And actually, many years ago when I was at Penn State, I did a journal club on a really cool paper which described this phenomenon called chromothripsis, which is like fancy nerd speak for chromosome shattering. Right. So in many cases of cancer, not all of them, but you can find chromosomes that have basically been shattered and just stuck back together, however which way, as like a Hail Mary, like let's see if we can fix this before we die kind of deal. And although the cells often survive, that can in turn quite often lead to cancer because you're messing up a lot of the control of various different genes and proteins when you just randomly stick things back together again, right? So that's another way of looking at this whole process kind of like turned on its head. You can fix those double-stranded breaks, but if you have a lot of them, there's no guarantee you're going to stick the right end to the right end. The only way to do it perfectly is through homologous recombination. There you go. Anybody got any questions? Quite a lot to cover in one class, but hopefully it all made sense. Pardon me, if it didn't, please do feel free to email me any questions. Don't forget, we have next exam on Thursday. I'll be there for the beginning of the class, maybe the first 45 minutes, depends on how the coffee's going and if we've got interesting emails to reply to. If you have any questions about the exam itself, after that, just contact me by email and that'll help. The exam should, fingers crossed, if I can figure out how to use Blackboard properly this time, be available Thursday morning at the normal start time of our class. And it will be available until midnight. So you have the whole day, same as before, but hopefully without any like crazy power outages and snow on the ground. But if you have any questions about that, just let me know and I will be more than happy to answer them for you. Awesome. Well, have fun, study hard. I'll see you either tomorrow or maybe on Thursday.